Meet Dr. Lianne Trigiani. She's a postdoctoral researcher at Cornell University's Meinig School of Biomedical Engineering. In the Schaefer-Nishimura lab, she focuses on dementia research. My name is Pietro Michelucci, and I'm the executive director of the Human Computation Institute. At HCI, we run projects including stall catchers, where thousands of volunteers help scientists analyze blood flow in the brain in partnership with the Schaefer-Nishimura lab. A year ago, I sat down with Lianne to talk about her work. We began with her motivations to pursue research in the first place.
I was initially just interested in mechanisms of memory formation. So I was just curious, what is an actual memory and not this like philosophical thing? If it's what makes us who we are, or if this is, a bit of a romantic idea, right? It's like memories make us who we are, and then, okay, then how can I measure this, or what physically is a memory? So that really got me interested in just memory research. And then pretty soon thereafter, like a couple of years after that, then my grandma started to develop Alzheimer's disease, which is obviously very intimately linked to memory. And then I thought that might be a nice kind of way, not only to be motivated, but it would be a good use of my time, I would say, to try and make a difference. Because I could see, especially at that time, that was about, 10 years ago now. So and it took quite a while for it to develop. So she's otherwise a very healthy individual. So it was a long progression that we saw. It was just only a few years really into her diagnosis that I decided that I was going to dedicate a good portion of my life to.
Research has found that pure Alzheimer's disease is very rare, and it often occurs with other dementia-related illnesses, especially related to blood flow. According to Lianne, inflammation in the brain, which is an immune response, can precipitate Alzheimer's disease. That said, for most people, Alzheimer's risk is not determined by one thing. It can be shaped by a combination of genetics, health, and environment.
What I'm referring to mostly in my research and what I'm trying to look at is late onset dementia. And this is where we don't have a clear-cut idea of the cause of it. And it's usually a combination of risk factors, some of which are genetic, but they are, just because you have a particular gene doesn't guarantee, like these other genes.
APOE is a gene that everyone carries. Some people have APOE variants that are more likely to contribute to Alzheimer's disease than others, in part due to factors like how the gene influences inflammation, responds to aging, and interacts with blood pressure.
So like the APOE gene that we'll probably be talking about a little bit, this confers a risk depending on which version of it you have, but doesn't guarantee. So point being, like your risk score or your combination of risk factors that you have can potentially exacerbate a larger immune response that makes you, in the long term, worse off, essentially. So it's not like one thing that's going to give you Alzheimer's disease. It's typically a combination of many factors.
In other words, Alzheimer's risk builds over time. Genetics matter, but they interact with other factors. One piece of the puzzle is blood flow in the brain. In the lab where Lianne works, researchers study stalls, which are moments when blood flow slows or stops completely in capillaries, which are tiny blood vessels in the brain. The big question is whether preventing these stalls can protect memory. Mostly, Lianne does her research on mice, with findings that may one day be applicable to humans.
Okay. So basically, I started in the lab, fresh postdoc, ambitious.
A lot of Alzheimer's research focuses on amyloid beta proteins, which form sticky clumps that build up and eventually harden into plaques in the brains of people with Alzheimer's disease. But Lianne wanted to do something different and look more closely at other risk factors, like genes, blood pressure, and aging.
And I was interested, again, in kind of veering away from the typical amyloid models and looking at a different kind of risk factor model. So I knew that the lab had these APOE mice. And then the first question was just, okay, does this genetic risk increase stalls or not? So we were just comparing E3 versus E4 mice at the beginning. So typically people study aged mice, but when I came into the lab, we just had, 3-month-old mice.
This is key. By studying younger mice, Lianne was able to examine different risk factors.
So we said, okay, let's see if we can look at the stalling maybe even before cognitive deficits occur. And there was a trend for E4 mice to have slightly more stalls. but it was not significant. And we added quite a few mice at that point. So we said, okay, there's something there, but not quite. And they didn't have any memory problems. So we were like, okay, it's still interesting. Like nothing's really happening in the model, but what else can we add then to this model that can maybe trigger cognitive decline? And this is a little bit my background for my PhD. I did this as well, as kind of models with risks. And then, okay, we would add a high cholesterol diet, or we would add diabetes or things like this. So then I looked around in the literature and tried to see, okay, clinically, what is present in humans. I've always known in the back of my mind that hypertension or high blood pressure is one of these very important risk factors. And then sifting through cardiovascular risk and ApoE, particularly ApoE4 genotype, what is most strongly associated with Alzheimer's disease and causes memory deficits. And then what was coming up was hypertension. So I just, okay.
By studying young mice, Lianne could see genetic risk in isolation before memory loss or major brain damage appeared. Her early results showed that ApoE4 increased vulnerability but didn't cause memory loss on its own. To understand what might push that risk into real symptoms, Lianne looked to one of the strongest dementia risk factors in humans, high blood pressure, which is called hypertension. Hypertension puts constant strain on small blood vessels in the brain. When combined with ApoE4, which weakens repair and heightens inflammation, that strain can destabilize blood flow. That's why adding hypertension to the model led to dramatic changes in stalls, blood flow, and memory.
So basically we added hypertension in these mice, and then it was the kind of skyrocketed from there, basically. So we added hypertension to the E3 mice and the E4 mice. And then this is why there's so much data now. But anyways, so we basically found pretty excitingly that the stall rate seemed to double in the E4 mice with hypertension, but didn't really change in the E3 mice. And then we also saw this decline in blood flow, decline in memory, and we were like, all right, we have something here, and now let's try to see what's happening.
Today, Lianne continues to collect data to further explore whether or not hypertension is a major risk factor for late-onset Alzheimer's disease. New data could strengthen her findings.
So we're still putting in data in stall catchers to see if that winds up being the case, to verify that. But yeah, so there's still more data coming for that with very carefully curated stacks.
Great.
So we're curious to see if that holds true.
Lianne is also looking into interventions and therapies that could reverse the stalls, restoring blood flow and possibly memory. One mechanism is antiplatelet therapy. Platelets are small blood cells that can contribute to inflammation when they're overactive.
But we saw a decrease in stalls after even just 1 day of this antiplatelet. So it works very, very fast. It peaks basically in 15 to 30 minutes after you administer it. It's already approved, which is always nice.
These therapies impact microglia, which are immune cells in the brain. Lianne is going to look more closely at how they interact with capillary stalling.
Well, what do I expect to find? There's a few ways we're looking at it right now. I suspect, definitely not 100% of the stalls are going to have a microglia nearby. I am curious to see what types of stall have a microglia nearby, though. What we're hoping to be able to see with the data, we can kind of 3D model it, and we can actually see when there is a stall there, or at least in a vessel that was previously stalled, are the microglia processes. So microglia have this big cell body, and then they have these little arms that can actually enter the blood vessels. So it's potential that they're obstructing it in that way. So like we can see in a stalled vessel, are they sticking their arms down there? Is that causing the obstruction? What are they doing?
Ultimately, Lianne hopes that her research will lead to more therapies that address Alzheimer's disease.
But I think it would definitely point towards, I would hope, for pharmaceutical companies to potentially take up on this and say, hey, this is an important thing to target if we really wanted to develop something specifically for Alzheimer's disease. I think realistically, because of how complicated Alzheimer's disease is that it's going to be some kind of combination therapy at the end of the day and not just one thing that's going to fix everything. But yeah, I think it'll hopefully encourage more doctors to think about which type of antiplatelets they're prescribing. Maybe aspirin is the way to go, but maybe P2Y12 is also the way to go. So they actually can be taken. Sometimes for some conditions, they're prescribed both because they are working along different mechanisms, and people have reported better improvement in what they're trying to fix, essentially, by using both instead of one or the other. So it could be a way, basically more evidence to show that this is a way to potentially prevent Alzheimer's disease.
Alzheimer's and dementia-related diseases form a huge research umbrella. So Lianne hopes that other scientists will continue testing their own hypotheses so we can piece together the complex puzzle of this disease.
There are so many hypotheses with Alzheimer's disease in terms of what is really causing it. So I think it's really important to keep an open mind to that, because we tend to get attached to a particular hypothesis, and that can be dangerous. There's so many things that can be involved. So I would say just to at least be aware of all these different hypotheses, not to get too discouraged if yours doesn't work out. I think just my advice for somebody who wants to enter science in general is just to be aware. I guess this is something that I always warn undergrads of when they join the lab, is that they tend to be very good science students and they're used to being right all the time. And it's just not the case in research. So like, you can come in with as many good hypotheses as you want. At the end of the day, if you're not testing things, like, you don't want to be testing things that we already know. So like, you're not doing experiments to prove yourself right all the time. Like, the point of doing research is to discover new things, and you have to be wrong a lot of the time in order to get there. So just to be aware of that reality if they really want to go into research, because that can be really discouraging for people when they first start off.
Alzheimer's and other dementia diseases don't have a single cause, and they won't have a single solution. Lianne's research highlights how genetic risk, blood pressure, immune activity, and blood flow interact over time, long before memory loss begins. At HCI, we're proud to be part of this research process with the Stall Catchers Citizen Science Project, and we're grateful to every one of our volunteer catchers who help researchers like Lianne analyze real brain imaging data. If you would like to be part of this mission, please visit stallcatchers.com today.